So another way to control CDK is by adding or removing CDK inhibitors. So here you have the active form, CDK with a cyclin, but then the inactive form is when you have this inhibitory protein wrapped around it and rendering it inactive. So three types of CDK inhibitors that I want to talk about are P21, P27, and P16. P21 is regulated by the transcription factor P53, so that promotes, it controls the transcription of the P21 gene, which then results in the P21 protein. So what the P21 and P27 do is they act on the G1S CDK, which is what commits the cell to go into DNA replication, and then SCDK, which is what actually helps in replicating the DNA during S phase. So if you're suppressing both of those, these two proteins do that for different reasons. So P21 does it if there is DNA damage, so you don't want to spread the damage to other cells, and then P27 would block that during G1 because during G1 your cell is just growing and living its normal life. You don't want it to replicate its DNA at that point in time. Now there's also P16, which acts on G1 CDK. So remember, G1 CDK is important for getting through the restriction point, pretty much to be able to go from G1 into S phase. Now this one is, so if you have this inhibitory protein, and it's blocking it, so it'll keep the cell in G1. But often, this protein is going to be inactive in cancer cells. So if it's inactive, it's not going to be able to inhibit G1 CDKs, and therefore the cell will just continue going into DNA replication and have this uncontrolled growth and division.